Okay guys, today we are going to start chapter 5. This chapter is all in the cell membrane. It's one of my favorite chapters. I think everything fits very nicely together in this chapter, and hopefully we get everything to make sense on how it works. So, like I said guys, this is all about the cell membrane. The cell membrane is selectively permeable. What this means is it's going to let some substances go right in, others it's not going to let go right in. Others, it might not let them go in at all, okay? So some might get in easily, some might have to wait a little bit, some, yeah, you're not entering. So all different ones. Here is a picture of the cell membrane. Now, if you look, obviously this is an animation, this isn't like under a microscope or fluorescently dyed or anything like that. But if we look in the middle here, okay, the green is a non-polar region, and you can see where it's very densely packed with this green stuff. There's none of these red and white molecules coming through. But where it's less packed, we do get some of these red and white molecules coming through. That's water. So if there is a lot of these green non-polar regions, water's not able to pass through. If there's only a little, water can pass through. Not in a great abundance, just a little bit, but it does pass through. And you can see up here where the yellow balls are, the water is able to get up close and personal to them, but just the same over here, it's not able to get past into the green area. Okay, so we'll talk about this the whole chapter. So first vocab word, it's called amphipathic. What amphipathic means is we are going to have phospholipids, and these are going to make up our cell membrane, and those phospholipids are going to have both hydrophobic and hydrophilic regions. In other words, some areas of this phospholipid are going to be polar, some areas are going to be nonpolar, and that's what we mean by amphipathic. It has both properties. It's polar and nonpolar at the same time. Here's a picture of the cell membrane. Okay, you can see everything that goes on. Here is our phospholipids right in the middle there. These big purple things that go right through our cell membrane, those are going to be some membrane proteins. And there's different kinds of proteins. We'll talk about each one. Along with this, we have some different tags here. We have carbohydrate tags. You can see the Glycolipid here. That's another tag. But what these are going to help us do is help to locate things that need to get in to our cell, okay? And like I said, guys, we'll go over these in more detail, but in this zoomed in version of it, we can also see that there is cholesterol in our cell membrane, and we're going to talk about what cholesterol does for us. Okay, I will attach this video instead of showing it to you guys. It's just a video on the cell membrane. What I want you to get out of the video is that our cell membrane is actually very fluid. We call it the fluid mosaic model. Why we call it this is because it literally acts almost like a liquid. Now, on a microscopic level, if we're able to see the phosphate group and the fatty acid chain, it's not turning from a solid into a liquid. But picture this. Picture you and all your friends are in the pool together, okay? You're in a pool, and you have those little pool noodles. You know what I'm talking about? The little, you could like sit on them or whatever. But pretend you're holding a pool noodle, and you're just bobbing up and down, up and down, up and down with this pool noodle in your arm. This is actually how the phospholipids are, but while those, while they're bobbing up and down, they're also moving. So if Johnny is next to, I don't know, Kristen, okay? Johnny might be next to Kristen for 1 second. He's a phospholipid, okay? But the next second, he might go around Kristen and go over to Rachel or Judy or someone else, okay? Doug might be over there and he wants to spend some time with Doug. I don't know. I'm just making up names. But the goal that I need you guys to understand is that the phospholipids are not fixed. They're going to move around. They're going to switch their position inside of the cell. Okay. Now, let me draw you something here. on our phospholipids, we're going to have two regions of it. Okay. The part that is pointing to the exterior and the interior of the cell, which is, let me change my color here. Oops. Which is this region right here, these circles. They're pointing to the exterior and the interior. These are phosphate groups. And they are hydrophilic. Now these down here, these are our fatty acid tails. And there's two of them. And they are hydrophobic, meaning they do not like water. So this right here, this is our construction of a phospholipid. If you remember, this is a polymer of our lipids. If we go back to the macromolecule chapter. So we have this phospholipid. It's two parts, the phosphate, which is hydrophilic, and we have the fatty acid tails. There's two of them. They're hydrophobic. Together, we have a hydrophilic region and a hydrophobic region. This is why we say that a phospholipid or the cell membrane is amphipathic. The other thing I want to mention to you guys about the fatty acid tails, I know in the macromolecule chapter, we went over two different types of fatty acids, unsaturated and saturated. They can be either, okay? They can be either. So we can have, I'll show you the three different ones here. We can have some of our phospholipids, okay, with two, this is two, I'm going to say un for unsaturated. There. We might have this one here, two that are saturated, or we could have one of each. So you can see the difference between them is that the unsaturated ones are kinked due to that double bond being between our carbons. The saturated ones, they don't have that little kink in them, and that's because there are no double bonds in between our carbons. It's all single bonds. Okay? So there is your construction of a phospholipid. Let me draw one more thing for you guys. So if I have our cell here, just to elaborate on something I mentioned earlier, we say that the cell membrane is a lipid bilayer, bi meaning two. So if I were to take this little region right here, okay, take it out, we'll magnify it, okay? That's our magnifying glass. It's not a lovely ice cream stick with some nice vanilla ice cream wrapped in chocolate up top. I wish it were, though. But anyway, let me just enlarge that little area there. Okay, so right here, this would be outside of the cell. And in here, obviously, would be inside of our cell. Inside. Outside. Let me put my fatty acid tails in here. Two on each of our phosphates. Okay. This is what I meant to when I said earlier, guys. The phosphates are connected to the outside and the inside of the cell. Remember, they are hydrophilic. So you could see that the outside of the cell, this area right here, which there's water outside of our cell, along with like the gap junctions and the tight junctions we talked about last chapter, there's water out here, and that water can get up close and personal to our phosphates, both outside and inside, okay? But when the water tries to go through, if there's a lot of the fatty acids there, the water gets repelled backward and stays inside or up here outside of our cell, okay? So that's what I meant whenever I said the phosphates are pointing to the inside and outside of our cell. Okay. And here's everything we just drew, guys. You could see up here at the top, we would have this one, which would be our unsaturated, because it has that kink in it. And this guy right here, our straight one, this one would be saturated. Okay? And there's our picture we just drew. Last slide. Okay, so we already talked about this. The lipids, they'll move. Okay, the phospholipids, they're going to move. There's also going to be very large proteins embedded in our cell membrane. So they go straight through. They're embedded in our phospholipids, right in between them. Now, as the phospholipids move fairly quickly, the proteins are going to, move less quickly. Okay? What they did here is they took a human cell and a mouse cell and they intermixed them. Okay? Now, obviously, they're not trying to make a human mouse baby, but they mixed them and they found that the proteins actually mixed after an hour. Now, if it was phospholipids, it would have been within seconds, but you guys can see the bluish purple color and the pink color, they mixed, okay, and made one big cell. Okay, so this is a very important slide. We'll spend some time on this in the next few here. So as the temperatures cool, so it gets cold outside, okay? For you guys, hopefully your body is maintaining homeostasis, where the inside of your body, even though it might be 32 degrees outside, is not becoming 32 degrees inside your body. If that's the case, you're dead, okay? So we don't want that. But as the temperature cools inside your body, what's going to happen is our cell membranes are going to be less fluid. They're going to go from a liquid state to more of a solid state. We do not want this. We want our membranes to be nice and fluid, okay? So what can happen here? Let me draw a picture. Actually, I think it's on the, what can happen here is as our membrane actually turns from a liquid and starts to become solid, our membrane can actually separate right here, okay? So we have a lipid bilayer, one layer here, one layer here. And what can happen is if it becomes solid, it will actually, instead of being like this, it will separate from one another, okay? Yeah, that's not good, because then we don't have this nice lipid bilayer protecting our cells and regulating what goes in and out, okay? But what this tells us, guys, is that our area right here in the middle, that area, okay, since we are breaking right there, those bonds that are going to hold the one layer to the other layer are much weaker. They're much weaker than the bonds that are holding our phosphates, holding our phosphates to our fatty acids, or holding our phosphates maybe to our phosphates, okay? So the bonds in between our two layers are very weak, very weak. Now, if you guys look up here, we talked about unsaturated and saturated fatty acids two chapters ago. If we have unsaturated fatty acids represented by the kinks up here, our membrane is going to be more fluid, more liquid-like. If you remember, unsaturated fatty acids are oils. They come from plants. Yeah, we have some too in our bodies, okay? But they're liquids at room temperature. This one over here, we have a lot of saturated fatty acids. And this one is not going to be as fluid. So we want a good ratio of unsaturated to saturated fatty acids. Why we want this is to make sure our membrane is not too fluid, not too liquidy, but also not too solid-like, okay? Moral of the story, guys, is the more unsaturated, or you could say polyunsaturated fatty acids we have in our cell membrane, the more liquidy it's going to be. We don't want it to be too liquidy. Now, the other thing we're going to talk about down here is cholesterol. And you guys can see there, cholesterol reduces membrane fluidity at moderate temperatures, but at low temperatures it hinders solidification. So let's talk about what's going on here. Let's say, I don't know, we'll talk about fish. We have fish, and it's cold. They're like way, way north, okay? So cholesterol hinders or stops the membrane from becoming solid. So if the fish are really, really, really in a cold environment, what's going to happen is there's going to be more cholesterol coming to that area in the cell membrane to keep it from becoming a solid. Okay, so moral of this part, guys, or the big idea, is going to be cholesterol is going to help our membranes from becoming solid. It's going to keep them liquidy. Okay. All right. Oh, and also one more thing about this, guys. When the temperature is increased, just coming back up here, when the temperature is increased, so increasing temperature, it's going to go more liquidy, right? Yeah, so increasing temperature, it's more liquidy. Decreasing temperature, it becomes more solid. That's why we need the cholesterol in there. But as we increase the temperature, we're becoming liquidy. Those phospholipids have less interactions with one another because it's becoming a liquid. They're not as close together. All right, this is how normal things act. Water's the weird one that we went over in chapter 2. But everything else, as it goes from a solid to a liquid, the things will space out a little bit. All right, we talked about cholesterol already. Okay, so if you are becoming cold, internally, obviously, our cholesterol is going to help keep our membrane fluid. It's going to maintain that fluidity. All right. Okay. This one, guys, is just about adaptations of our cell membrane. Okay. We have evolved where we're able to live in various temperatures. And again, that's dealing with homeostasis, maintaining our homeostasis. Different proteins in our cells. We have integral proteins. Integral proteins are going to penetrate our hydrophobic region of our lipid bilayer. So they're going to be attached to our phosphates, but they're also going to penetrate the fatty acid tails. What does this tell us about integral proteins? Well, if they're being connected to our phosphates and also being connected to our fatty acid tails, it tells us that our integral proteins are also going to be amphipathic, just like our phospholipids. It's going to have some areas that are hydrophilic. Those are going to be the areas that are attached to the phosphate. It's also going to have some areas that are hydrophobic, and those areas are going to be attached to our fatty acid tails. Okay? If an integral protein goes all the way from one side to the other side of our cell membrane, from the outside all the way to the inside, we call it a transmembrane. It penetrates the whole membrane. All right? Last one on there, we're going to have peripheral proteins. These are ones that they're bound to the surface. They're not going to go the whole way through. Okay. All right, I will paste this video, guys. It's just a little animation of inside the cell membrane. Okay. You guys can see that in our transmembrane protein here, we're going to have two sides. We are going to have a N-terminus side, which is going to point to the outside of the cell, and then we are going to have a C-terminus, which is going to point to the inside of the cell. Nothing too exciting there. Okay. So these are some different things we're going to go over with our functions of our membrane proteins. Here's a nice picture of them. They've recently found that back in the day, guys, we thought our cell membrane was going to be a whole lot of phospholipids, and we just sprinkled in a few proteins. Okay, we found out that our proteins are very, very specific, just like enzymes are with the specific substrate. Since we found that they are so specific to things they are going to let pass through these proteins, we found that there's a lot more proteins than we originally thought. Okay, so there is a lot of phospholipids, but there's also a whole mess of proteins to go along with it. All right, so different tags we have outside of our cell and inside that are going to let things pass through. Guys, these things don't have eyes, okay? You can't think of them being able to be like, you know, oh, hey, here's some water, I see you. No, they can't tell where the water is. So we need these tags in order to help locate what we're going to let in and out of our cell. Okay. So what we're going to have is we're going to have these carbohydrates. They are going to be bonded to either lipids or proteins. If a carbohydrate is bonded to a lipid, it's called a glycolipid. If it's bonded to a protein, it's going to be called a glycoprotein. And this is going to help with cell-to-cell recognition, recognizing other cells or recognizing things that may want to enter the cell. Okay. So in our membrane, guys, it just says we have distinct inside and outside faces, okay? One's going to be on the inside, one's going to be on the outside. What this is going to do is, once again, help with the carbohydrates will help us with knowing what's going to come in, what's going to come out, all right? And you guys remember our pathway of a protein from the previous chapter. We went from the nucleus to the rough ER to the Golgi, with vesicles transporting things in between, and then out of the cell. Once again, our little tags are going to help us for that vesicle to fuse to the cell membrane and allow things to exit our cell. All right, we're going to do a little bit of the next section today, and then we will call it a day. So, hydrophobic, hydrocarbons. We learned already from chapter 2 that hydrocarbons are hydrophobic. These are things that are going to just be made of hydrogen and carbon. So if a molecule is made of only hydrogen and carbon, it's always going to be nonpolar. It's when we start bringing other things into it, like oxygen molecules, all right, or a hydroxyl functional group or different functional groups. This is what's going to make them polar. Now, nonpolar molecules that are hydrocarbons, or things just like small things like oxygen gas, they're going to be able to pass through the cell membrane really easily. They can go straight in between the phospholipids. When we start adding other things to make them polar, that's when they are not going to pass easily. Okay? So polar molecules like sugar, water, anything that pretty much dissolves in water, they're not going to be able to go right through our phospholipids. This is when we are going to bring our transport proteins to allow these polar, these hydrophilic substances into or outside of the cell. There is a special one for water. It's called an aquaporin. So there's a lot of substances out there that we're going to be letting in and out of the cell. Water is the only one that has a special name for it. And again, it's called an aquaporin. You get your aqua there, okay, your prefix. There's two different types of other proteins. There's channel proteins and carrier proteins. The channel proteins, as it says here, we're going to let hydrophilic things or molecules and ions through. They're just going to slide right through our channel protein. Carrier proteins are a little different. Yeah, it's still going to be letting ions and things through, but what's going to happen, instead of the things going, like in the channel protein, going straight through, what's going to happen is it's going to be like this, and something's going to be dropped in, so now it's caught in there, and then the protein is going to change its shape. So if this is outside of the cell, we just dropped my pencil into my protein from the outside of the cell. What will happen is it'll close on this side, it'll open on the other side, allowing the pencil to come into the cell. Okay, easy enough. Sorry for the noise. But that's the difference between a channel protein and a carrier protein. Channel proteins is just one pathway right through. Carrier proteins, they're going to change their shape in order to let something into or out of the cell. Okay, we will get into diffusion next time, guys. Have a good rest of your day, and I'll see you tomorrow.